Yeah. It is my privilege to welcome you all to this fourth memorial lecture held in honor of our founder, Dr. Kallam Anji Reddy, whose remarkable contributions to society, science, and sustainability have left an enduring legacy. We are deeply grateful to you for your presence and look forward to an engaging session that reflects the spirit of inquiry and excellence. Thank you for joining us in paying tribute to a life devoted to science and the betterment of human health. I now request the Avasa Security Manager, Mr. Jabir Hussain, to brief us on the safety protocol.
Good morning, everyone. My name is Jabir Hussain. I am the Security Manager of Avasa. I am here to brief you about security briefing how to handle an emergency situation and how to respond in case of any emergency. We are at the first level at Constellation Hall. In case of any emergency, we have fire exit doors. All the exit doors are placed with the fire signages. Please follow the exit doors and signages to reach to the safe assembly point, which is near to our main gate. For medical emergency, we have near attached PACE hospital, and we have all the equipments like wheelchair, stretcher, and AED to respond with the medical emergency. For your information, today we have not scheduled any mock drill in hotel. If you hear any fire alarm, please inform our colleagues who all are here to assist you. They will call our in-house trained DMT team members, disaster management team members, to handle the situation. Thank you.
Yeah, thank you. May I now request Mrs. Deepthi, Mrs. Anuradha, Mr. Satish Reddy, Mr. J.V. Prasad, and Ms. Shravya to kindly come forward and light the lamp. Thank you. May I now request Mr. J.V. Prasad to welcome all the distinguished guests.
Good morning and a very warm welcome to all of you gathered here on a Sunday morning. Thank you so much. Many of you in the audience have worked with Dr. Reddy, and I meet you here every day, and it's such a joy to see you all. And we are deeply honored by your presence. It means a lot to us. We're also deeply honored to have with us today Professor Guido Kremer, a globally acclaimed leader in aging, cancer research, and immunology from Université de Paris, France. I hope I got the name right. This lecture series began in 2023 to celebrate Dr. Reddy's life, legacy, and passion for science. Every year, we have been privileged to host some extraordinary scientists whose work embodies cutting-edge innovation, translational impact, and breakthroughs poised to redefine tomorrow's medicine. At the heart of his vision was deep science, driving affordable and accessible medicines for everyone. A true scientist at heart, he carried into business the same relentless drive for novelty, innovation, and excellence that really defines scientific discovery. He had this quote on his whiteboard, and it's been there for many years, many more years than I can remember. And he drew inspiration from this quote from George W. Merck. We try never to forget that medicine is for the people. It is not for the prophets. The prophets follow, and if you have remembered that, they have never failed to appear. The better we have remembered that, the larger they have been. And this philosophy guided Dr. Reddy's journey from making medicines affordable in India to pioneering technology that helped establish our country as the pharmacy of the world. His contributions to this achievement were very significant. And he left an indelible mark on all who knew him. From him, we learned to embrace the magic of science, think boundlessly, rally teams around societal goals, and pursue solutions for unmet medical needs through relentless innovation. And he believed that with the right vision, resources would follow, and they are never a constraint. And our history has kind of proved him right. He also encouraged us to embrace a broader responsibility to care for patients, people, and the planet. While he was not explicit about sustainability, his emphasis on holistic impact inspired us to make it a core pillar for us. Today, we stand among the leaders in sustainability, reflecting the enduring values he embedded in our culture. These three elements, science, society, and sustainability, form the very theme of this lecture series and also remain woven into the fabric of our organization. And our milestones in complex generics, new markets, drug discovery, and emerging technologies all go back to this scientific ambition that he ignited amongst us. Through this annual event, we hope to inspire the pursuit of science for the greater good of society as well as sustainability. We seek to connect world-renowned minds like you with India's scientific community, fostering bridges between academia, industry, and the healthcare systems. And nothing delighted Dr. Anji Reddy more than engaging with fellow scientists and sparking such collaborations. He was at his happiest in the company of great scientific minds. He was endlessly curious, always eager to understand the frontier directions in medicine, the emerging breakthroughs in translational research, and how fundamental discoveries would translate into real-world cures. There is no finer tribute to him than an event that brings together distinguished scientific leaders like many of you in the audience to inspire and connect. We're again truly grateful to Professor Guido Kramer for accepting our invitation, especially during these uncertain times where travel is quite a uncertain thing. And his pioneering contributions to programmed cell death, autophagy, cellular metabolism, immunity, and cancer therapy have fundamentally reshaped modern oncology, particularly our understanding of treatment, response, as well as resistance. He has many honors to his credit, and some of them are the Leopold Griffel Prize, the International Prize, Lombardi and Research in 2019, the Mark Foundation Cancer Research Aspire Award, and the ERC Advanced Investigator Award. Professor Kramer leads influential research at INSERM and Gustav Rossi Comprehensive Cancer Center. His work bridges basic biology and clinical practice in service to society, a perfect alignment with the spirit of this event. He's frequently ranked among the most cited cell biologists globally, specifically for cell death and mitochondrial research. He has authored about 1,600 articles, including numerous papers in top journals like Cell, Nature, and Science. Professor Comer has founded or directed several research institutions and units in France. These institutes span a wide breadth of scientific pursuits from fundamental cell biology to translational cancer research. We are also delighted to welcome Dr. Guru Reddy, a friend of ours, founder and chairman of Continental Hospitals, Hyderabad. He's a distinguished gastroenterologist trained at Baylor College of Medicine in Houston, Texas. And Dr. G.N. Reddy built one of the largest GI and liver clinics in Houston. He's widely regarded as an icon in his field and a masterful institution builder. Bringing his vision of high-quality, patient-centered care from Houston to Hyderabad, he established state-of-the-art Continental Hospitals in Hyderabad. It's a pleasure to have you with us today, and we look forward to your insights and memories from your interactions with Dr. Reddy. And with grateful thanks, I welcome all of you again. And many of you have joined online, thank you. This is our humble way of honoring the scientist, entrepreneur, and philanthropist, Dr. Kallam Anji Reddy. Thank you very much.
Thank you very much, Mr. Prasad. And as you said, Dr. Anji Reddy's legacy continues to inspire generations of scientists. So we will now play a video as a tribute to Dr. Anji Reddy, a visionary scientist, entrepreneur, and leader.
What really drives me is the science. That can I finally bring a drug into the market which can change the way people live? Whenever I think of my father and mother, there is one picture that comes to me invariably, is that my father would go to the Vijayawada, which is just about 2 miles from our place. All you have to do is walk across the Krishna River. And he would go and buy some Ayurvedic roots and few other dried fruits kind of things. Mm-hmm. He would bring them, and my mother would powder them. Mm. In spite of the fact that from morning 5 o'clock till night 8 o'clock she is hardworking, in spite of that she always found time to do this. And the powder she will hand over to my father, and he will sit there. I cannot forget that scene, you know. He would take the powder and mix with something and make it into pills. Okay. So this ritual I have seen with my own eyes, and my mother herself told me this, that your father had this principle that we will always give it free, we will not charge. Because for us, God has given this land, our job is to grow turmeric, grow dal and all that, and sell, and out of that, if we have to give to some poor people, we will give. But for the knowledge that I have gained, I will not charge. It was a dream. It was not a plan on the drawing board, no. But it was a dream. I want to be a pharmaceutical manufacturer. And obviously, God willing, maybe number one in India. If I didn't have a vision at various stages in the evolution of this company wouldn't have reached this stage. You should not think that because of failure we should cut corners or anything like that. The vision should continue in spite of the failure. Because if there is that element in you which is better than many other people, you can be one of the top companies. This foundation came out of my anger. I read in Newsweek about the poverty of India, and he wrote a sentence which hurt me so much. He said, except that certain regions of India are worse than sub-Saharan Africa. I was so angry. Say maybe for after 10 years or so, if I take my own plane and go around and see patients who are on my drug, that is the greatest satisfaction in life. And they will, mind you, there will be millions.
Thank you. May I now request Dr. Srinivas Oruganti, Director of Dr. Reddy's Institute of Life Sciences, to introduce our distinguished speaker for the memorial lecture.
It's indeed a privilege to have Professor Guido Kromer from University of Paris to deliver Dr. Kallam Anji Reddy Memorial Lecture today. Mr. Prasad has given some of the significant achievements of Professor Kromer. I will share a few other details. He's the Director of Metabolomics and Cellular Biology Platforms at Gustave Roussy, and he heads the INSERM team Metabolism, Cancer, and Immunity. He's also a professor at the Faculty of Medicine at Paris Descartes University, an adjunct professor at the Karolinska Institute in Sweden, an honorary professor at the Suzhou Center for Systems Medicine in China, and the Deputy Director of Cordelius Research Center, again in Paris. Professor Guido Kromer has made seminal contributions in the last several decades, particularly in the area of cellular biology and cancer research. He has elucidated the mechanisms of mitochondrial control in cell death, the anti-aging role of autophagy, immune responses that essentially ensure the success of cancer treatments. As Mr. Prasad has mentioned, Professor Kromer has published more than 1,700 publications with a significant number being in the CNS journals, Cell, Nature, and Science, Science Translational Medicine. He also serves on the several editorial boards, and he was editor-in-chief of journals Cell Stress, Microbial Cell, Medcom Oncology, and seminars in immunology. He has received several prizes and accolades, some of which have been again mentioned. But let me just give you a kind of a small split across the different countries in Europe, starting from Belgium, Baillet-Latour Health Prize, from France, Prix d'Esquin, Prix Leopold Griffel, Grand Prix Ruben Rose. In Spain, International Prize for Oncology and Ramiro Carrigal and Bull SE Award. From Switzerland, the Brubacher Prize, and the European Union-sponsored Descartes Prize, as well as most recently the very important Italian prize, Lombardia and Researcher Prize. He has also received 2 European Research Council Advanced Investigator Awards, where he has led teams across multiple countries on these projects. One of the outstanding contributions to Professor Kromer has been also to build the entire ecosystem. He has actually been the editor-in-chief for 6 journals, Cell Death and Stress, Cell Stress, started the journal Oncoimmunology, Medcom Cancer, Microbial Cell, and is part of several very prestigious academic societies like Austrian Academy of Sciences, Academia Europaea, European Academy of Sciences, European Academy of Sciences and Arts, European Academy of Cancer Sciences, and several others. In particular, he has been the founding president of the European Academy of Tumor Oncology, as well as the president-elect of the European Network for Cancer Immunology. Professor Kromer will be speaking on anti-aging and the science and what governs the pathology, and potentially may share with us today in Hyderabad about how some of these things can potentially be addressed. But with his approval, just some time back, I wanted to share 3 very popular terms that he has coined, which actually gave us a very deep understanding about this entire immuno-oncology field. One is immune cell death, and the second one is calorie restriction mimetics. And the third one, which is certainly going to be a very popular term in the future, is carbotoxicity. I take the liberty of mentioning these 3 because he will be speaking on the different topic. With that, again, Professor Kromer, it's indeed a privilege. We're very grateful to have you here. I warmly welcome and request you to please deliver the Dr. Kallam Anji Reddy Memorial Lecture. Thank you.
It's a great honor and a great pleasure to be with you. Thank you for having me. I found a citation by Anji Reddy that I liked very much because it reflects what actually science, biomedicine, pharmaceutical development should be about, which is the humanistic aspect of doing research, developing drugs for others, for the sake of humanity. And I will not read the entire quote that you certainly know, but I like some words in this quote which are part of my everyday lifetime, which is ecstasy and exaltation. These are words that are not usually used in the rational discourse that we have, but that actually feed us and make us, keep us going. So it's an immense pleasure to follow in the footsteps of this giant and to give a memory lecture in his honor, Anji Reddy. And I am very much pleased that we have the family members here and also all the disciples, as I may say so, who are following the work ethics of Dr. Reddy in his institute and in this spirit here. So I will talk about a tissue stress hormone that you probably never heard about, which drives pathological aging. The last project that I have been conducting in our laboratory. Here are my disclosure information. And I actually start here with some reflections on the frontier between health and disease. So in my laboratory, I like to ask impossible questions that lead to impossible answers. And so one of the questions was, what are actually the principal characteristics of health? Not in the medical sense that we always understand health as the absence of disease, but in the sense of the organizational features of living beings that define the healthy state. And so if we focus on the center of this slide, there are some organizational characteristics such as spatial compartmentalization, maintenance of homeostasis, and adequate responses to stress that allow the maintenance of the healthy state. And so if we just think about the integrity of barriers and the containment of perturbations, it appears quite obvious that the many internal and external barriers that we have in our organism, ranging from the barriers that are around our organelles, the barriers about our cells, the barriers that separate our organs, or the skin barrier, have to be kept in an impermeable state to allow the healthy state to continue. And similarly, any perturbation that occurs locally across our body has to be contained in space and in time to avoid excessive and pathogenic reactions. And when we move to the maintenance of homeostasis, one of the essential features of our organization is that our building blocks have to be turned over, destroyed, and rebuilt to be recycled. And this occurs with entire cells that die and are renewed, or with organelles within the cells that are usually destroyed by autophagy, a self-digestive mechanism that is piloted by lysosomes, and then renewed. So keep the cells alive and striving. And even macromolecules have to be turned over, as is here illustrated for proteins that are denatured and renewed. And homeostasis is also maintained by the organization of our body. So there are different layers or strata that range from molecules to organelles to cells to organs, organ systems, and the meta-organism that includes our body plus the microbiota that colonizes it, the gut. All these layers are integrated in circuitries, and in each of these layers, the hallmarks of health have to be present under a continuous fluctuation dictated by time, the best known fluctuation being the circadian rhythm. And if we think about responses to stress, of course, we know that perturbations occur all the time. And the maintenance of the healthy state requires the repair or avoidance of the perturbation to allow for complete recovery, which can occur in two different fashions. One is just to return to the normal state, or a better option is actually to desensitize the system and to render it more resilient to the stress that has been overcome, a mechanism that's called hormesis, and that allows us to educate our organism towards higher resistance to stress. And all this involves many, many mechanisms that we have no time to talk about, among which stress mediators play a major role. And one of these major mechanisms is, of course, homeostatic feedback loops, in which an input, a perturbation, leads to a response that causes an output which ideally closes a feedback loop to remove or limit the perturbation. And unfortunately, in pathology, often these feedback loops get astray and are converted in pathogenic feed-forward loops, meaning that the output amplifies the derangement or perturbation. And in this case, if the response involves a molecule that we can measure, it becomes a biomarker of perturbation and pathology, and even an actionable biomarker, meaning that if we act on this response, we can improve the properties of the system. And so one of the questions that we have been asking, at least philosophically and then experimentally, is how is stress and perturbation communicated through the different layers that compose our organism, the strata, and through the different mechanisms that maintain health. Just think about 1 second. One single amino acid changed in one protein, let us say in cystic fibrosis conductance receptor, CFTR, which causes cystic fibrosis, one single mitogen in one molecule is sufficient to cause perturbations that go to all the strata. So there's communication. Or when we think about the microbiota in the gut, the simple dysbiosis, or an overcoming of pathogenic bacteria, leads to systemic disease all over through the different layers of the organism. And so we know that stress hormones play a major role in this communication of perturbations through the organisms. You all have heard about catecholamines, adrenaline, noradrenaline. You have heard about glucocorticoids. These are the official known stress hormones that are extremely important for the acute adaptations to stress and for the so-called fight or flight response to external responses, predators, for instance. But they are also immensely pathogenic. And so this is perfectly illustrated by the fact that there are multiple drugs that block adrenergic receptors, which are the receptors for catecholamines, adrenaline and noradrenaline, or antagonists of glucocorticoid receptors that are used in the routine management of major age-associated frequent diseases. And you have on the slide hypertension, prostate hyperplasia, all sorts of cardiovascular diseases, heart failure, cancers, and metabolic syndrome in all its facets. So stress hormones can be pathogenic if they are chronically elevated. And so we started our question on new stress mediators with the following reflection. Cells, and the core business in which working is cell biology, can adapt to stress and then just allow for organismal homeostasis, but they can also be eliminated by stress, either by a permanent cell cycle arrest, which is called senescence, or eliminated by cell death. And what we were wondering about is how is this communicated to the rest of the organism, thinking about danger signals that can come from stressed cells as they adapt to stress, or can come from senescent or dying cells, and then affect systemic adaptive responses to allow or not organismal homeostasis to occur. And so we spent some time looking for this missing hormone desperately. And actually, the illumination came when I was reading papers that apparently have nothing to do with medicine. And so these were 3 papers in Journal of Cell Biology and PLOS Genetics dealing with a protein that is secreted by unicellular yeast species in response to nutrient scarcity through an autophagy-dependent pathway. And so it makes no sense that yeast would secrete a protein in response to hunger, unless that protein would have a function outside of the cells as a messenger. And we were also intrigued by the fact that the knockout of the gene coding for this protein, ACB1, was increasing lifespan in yeast. So I started to work with my group on the human or mouse equivalent of this protein, which has 2 names. ACBP stands for acyl-CoA binding protein, which describes the intracellular function of this protein, which binds to activated fatty acids, participates to lipid metabolism. And DBI stands for the extracellular version of the same protein, the same molecule, diazepam binding inhibitor, also called endozepin, that is released in response to stress and passively leaked from cells as they die. And actually, that protein displaces diazepam from its receptor. Diazepam is a benzodiazepine. The receptor is GABA type 1 or type A receptor in a specific version of alpha-1 and gamma-2 chains outside of the central nervous system. And for that reason, actually, that protein has a name that is rarely used. It is endozepin, which stands for the endogenous equivalent of benzodiazepines. It is widely expressed in all tissues by many different cell types, so it's not a classical hormone, it's a tissue hormone. And it is phylogenetically conserved because it is found in yeast, in plants, in all sorts of fungi, in all animals. And hence might have an important function because it's the trunk of the phylogenetic tree. So we found that this protein, also called endozepin, is expressed by all sorts of parenchymatous and myeloid cells in all tissues, transactivated by stress-responsive transcription factors, has intra- and extracellular functions, is actively secreted from stress cells by an autophagy-dependent pathway or passively leaked from dying cells, acts as a neuropeptide on GABA-A receptors, mimicking benzodiazepine effects, and actually plays a central role in what we call the hunger reflex. So in all sorts of species, the nutrient scarcity leads to the stimulation of food-seeking behavior or appetite, which depends on this protein. And so here you have some examples. So you find it in slime molds, in different yeast species, fungi, in all sorts of animals in which systematically this protein stimulates different kinds of food-seeking behavior. So in yeast, it will be sporulation. In C. elegans, it is pharyngeal pumping. Or in mice, it is just looking for food and eating it. And so the knockout of ACB-DBI, so its inhibition by genetic tricks, stimulates autophagy. So ACB-DBI is an endogenous inhibitor of autophagy. And if we knock out ACBB, we find an increase in lifespan. So in chronological lifespan experiments, the red line for the knockout compared to the black line, which is the control, the wild type, there's an extension of lifespan by 100%. That is lost after simultaneous knockout of autophagy, essential genes for autophagy, such as ATG5 or ATG7, meaning that we can say that The inhibition of ACVDBI increases longevity in yeast because it stimulates autophagy. And so in yeast, we did some work to identify the typical rule that is actually valid for all molecules in biology, which is called antagonistic pleiotropy, meaning that the same molecule can have physiological and pathological effects, bad and good effects. And so the adaptive response to food scarcity in yeast, which is sporulation, so yeast travels in the air when it is hungry to find another source of food. That response, it depends on ACVDBI, but in yeast, ACVDBI also has a maladaptive function because when it is expressed, it favors chronological aging and cell death. And we find exactly the same in mammals. Antagonistic pleiotropy, meaning that there is an adaptive function, which is the response to food scarcity or cold stress that leads to the release of ACVDBI from cells through an autophagy-dependent pathway to act on GABA-A receptors and then to stimulate food intake and anabolic responses. And it also has a maladaptive function, meaning that in response to chronic stress, ACVDBI is released from stressed dying cells to act always on GABA receptors, always to inhibit autophagy, but because autophagy is a major adaptive response in cells and an anti-aging mechanism, this favors then lipoanabolism, inflammation, senescence, and cell death. And so I give you here an example of the importance of ACVDBI in human and mouse studies. For instance, ACVDBI is deficient in people, adolescents usually with anorexia nervosa, and you actually can predict from the initial plasma levels of ACVDBI whether a patient will relapse after treatment for anorexia nervosa or not. And in mice, you can use ACVDBI as a treatment for anorexia. So if you induce anorexia by chronic stress or by chemotherapy, you will induce normally a reduction in food intake and body weight, osteoporosis and sarcopenia. But if you provide ACVDBI by genetic or pharmacological tricks, you prevent the manifestation of anorexia with these side effects. And this is actually not an effect of ACVDBI directly on the central nervous system. It's an indirect effect on metabolism and on LCN2, which is one of the proteins that can cross the blood-brain barrier, which is not the case for ACVDBI, but is the case for LCN2. And then ACVDBI can stimulate orexigenic effects to stimulate food intake and to counteract anorexic signaling. And ACVDBI also has maladaptive functions that I will dwell on in a second. So in humans, we see that with age, ACVDBI plasma levels increase in a linear fashion. So this is an example of centenarians. So if centenarians are in relatively good health, living in a nursing home without major pathologies, they already have 3 times higher levels of ACVDBI than normal controls, healthy under 50. And when these centenarians are hospitalized due to end-stage disease, ACVDBI levels ramp up further, meaning that we can say that there's an association between ACVDBI in the plasma and aging, and an independent association with disease. And so when we look at the proteomics of the plasma of these patients, irrespective whether they are hospitalized or in a nursing home, we see correlations between ACB-DBI and specific factors, including VEGF-A, which is a typical biomarker of senescence, of the senescence-associated secretory phenotype. And if we look at thousands of patients, this is a cohort of a Canadian cohort of people infected by COVID that have been hospitalized, we see that dozens of different markers of senescence, so cellular aging, or the so-called SASP, the senescence-associated secretory phenotype, which is a pro-inflammatory signaling pathway, correlate very strongly with ACB-DBI. So there's apparently an association with aging, and this has been found now in, just to that you can understand how to read this, like these small red points indicate the number of studies that have been done. So there are 18 independent studies that have correlated ACB-DBI in humans with aging, measured in years, in more than 150,000 patients. And you also see a positive correlation of ACB-DBI with body mass index, glycemia, inflammatory biomarkers, signs of dyslipidemia, and organ-specific disease biomarkers such as cardiovascular parameters, including troponin T from the heart or arterial stiffness, renal dysfunction, and liver dysfunction. All these are just biochemical markers, so we consider them as a continuous value that we correlate with ACB-DBI plasma levels, but we can also look at it in a different way. So here we are dealing with 5,000 different proteins determined in 1,000 different patients with COVID-19, and we find this association of ACB-DBI with different proteins that can be bioinformatically determined for their origin, for the organ of origin. And you see, for instance, that there are many proteins from the pancreas and the muscle that positively correlate with ACB-DBI plasma levels. And in the mouse model, you can induce pancreatitis and then see that ACB-DBI plasma levels go up. Or you can induce rhabdomyolysis, so destruction of the skeleton muscle by local injection of glycerol, to see that ACB-DBI plasma levels go up transiently as well. Just to show that lesions affecting different organs can increase ACB-DBI in the animal model as we see it in the patients. And we can also diagnose diseases as a binary category, yes or no, a categoric variable, and correlate the presence of disease with an increase in ACB-DBI plasma levels. So we see this for systemic diseases such as metabolic syndrome, type 2 diabetes, cardiovascular disease, inflammatory and autoimmune diseases, and organ-specific diseases affecting the lung, the kidney, the liver. Cancer is an aggregate of different types, is correlating with ACB-DBI plasma levels, and even neurological levels, and of course, morbidity and frailty. Why are there so many non-specific associations between ACB-DBI and diseases? Well, if you think about it for a second, most diseases, perhaps all diseases apart from psychiatric diseases, are associated with an increase in cell stress and death, and this explains the association. What is even more interesting, if we measure ACB-DBI plasma levels in apparently healthy individuals with no disease, and these levels are higher than expected for the age and body mass index of that person, that individual is at higher risk to develop future disease. And so they have found this for mortality for over 10 years after blood sampling, and also for frailty, future cancer diagnosis, and future cardiovascular events. And this is the first study that we published 3 years ago. So it is called DESIRE. Patients were involved in a nutritional study in the Loire region in France. No apparent disease. We sample the blood, and then we have a 12-year follow-up, and we see that future cardiovascular events, such as infarction or heart failure or stroke, are preceded by an elevation of ACVDBI plasma levels. And we have much more data in cancer. So if you look at heredity cancer predisposition syndromes, such as the p53 mutation in the germline that leads to Li-Fraumeni syndrome, practically all patients develop sarcomas and carcinomas at the age of 45. And we look at them before they have cancer, and we see an elevation of ACVDBI plasma levels that actually can be used to diagnose Li-Fraumeni syndrome. And we see similarly in women that bear mutations in BRCA1 or BRCA2, before they develop breast cancer or ovarian cancer, they already have higher ACVDBI plasma levels than the controls. And this is also found in non-syndromic cancer patients. So here we are dealing with an enormous study of 13,000 patients that were also enrolled in a nutritional study with a very long follow-up, more than 8 years. So we identified during follow-up some 700 patients that would develop cancer, and then measured the ACVDBI at the time of enrollment in this study, compared to controls that were matched for sex, age, body mass index, and all known cancer risk factors. And again, find a slight increase in ACVDBI plasma levels that is statistically significant and particularly impressive for the future lung cancer patients. And so we confirmed this result in patients at risk. So these are patients that are heavy smokers, that have not yet developed cardiovascular disease, but are at risk of doing so because they have dyslipidemia or coronary calcifications. And you see that these patients, when they develop lung cancer in the future, have higher ACVDBI levels at enrollment of the patients than the controls, or the patients that have developed no lung cancer. And actually, if you split the cohort according to the median ACVDBI plasma levels, you see that those who have high levels in red will have a higher risk of developing lung cancer for at least 12 years than those who have low ACVDBI plasma levels, which is very impressive. So what are we doing with this? So the game that we are playing in our laboratory is that we identify human diseases in which ACVDBI plasma levels are elevated. This is true for all this spectrum of different diseases. Apart from Alzheimer's, where the elevation of ACVDBI is in the cerebrospinal fluid, not in the blood, because ACVDBI does not cross the blood-brain barrier. And so we go through all these diseases. There's association. We try to establish causation by doing studies in mice. And so what is written here in red, response to inhibition of ACVDBI. We're talking about infection, septic shock, cardiovascular disease, including infarction, heart failure with preserved ejection fraction, the entire spectrum of liver diseases that you can imagine, from steatosis to fibrosis, cirrhosis, and hepatocellular carcinoma, kidney disease and failure, cancer, which is involving ACVDBI-mediated immunosuppression, eroding immunosurveillance, and hence increasing cancer susceptibility, osteoarthritis, neurodegeneration, diabetes, immune dysfunctions in the context of septic shock, and metabolic syndrome induced by glucocorticoids, or Cushing syndrome. And we also have an effect on senescence, measured as by senescent markers in many different organs, and I will mention this briefly. So one of the most spectacular results that we obtained was actually in Cushing syndrome. The Cushing syndrome is a spectacular disease. I say spectacular, fortunately it's rare, but it's spectacular because you can see, you can diagnose a patient with Cushing syndrome from 10 yards of distance. It is caused by glucocorticoids produced by tumors or in response to tumors that produce ACTH by the adrenal gland, or it is produced by high-dose glucocorticoid treatments as an iatrogenic phenomenon. And so you see this transformation of that person with a redistribution of the lean mass towards the fat mass. So you have a visceral adiposity, you have the fat built up behind the neck, the so-called buffalo neck. You have atrophia of the skin adipose tissue, so it's a lipodystrophy. You lose muscle, sarcopenia, you lose bone, osteoporosis, and you have skin abnormalities, depression, and metabolic syndrome with hyperinsulinemia, insulin resistance, dyslipidemia, and so it's a very dramatic phenotype that can be lethal due to cardiovascular disease or infection. And so we all thought that Cushing syndrome is due to the action of glucocorticoids and glucocorticoid receptors that are expressed in many different cell types. And as a matter of fact, it turned out that it's much more complicated. Glucocorticoids act on hepatocytes so that they secrete ACVDBI that then acts on GABA receptors to inhibit autophagy, and this is the mechanism through which Cushing syndrome manifests. If we inhibit extracellular ACVDBI with antibodies, hence immunological tricks, or if we knock out ACVDBI in the adult mouse by inducible techniques, or if we mutate the ACVDBI receptor, the GABA receptor in the specific subunit, GABA receptor gamma 2, we completely abolish all manifestations of Cushing syndrome in a mouse model. And we do not only prevent Cushing syndrome, we even can treat Cushing syndrome. So this is the scheme of an experiment in which mice are fed first for 3 weeks corticosterone, which is the cortisol equivalent in mice, and then they receive a control antibody or the ACVDBI neutralizing antibody. You see that from week 3 to week 6, the antibody treatment will normalize the food intake, normalize the body weight, and actually also have an enormous impact on the biochemistry. So, for instance, in Cushing syndrome, the second group compared to the first, the control, you see changes in glycemia, insulin levels, free fatty acids, triglycerides, cholesterol, and so on, that are reversed and normalized by ACVDBI neutralization. And actually, if you look at it from a mechanistic point of view, the prolonged glucocortic exposure that has enormous effects on the body composition and on the function of the body, that mimics accelerated aging with metabolic syndrome, osteoporosis, immunosuppression, cardiovascular disease, and even neurodegeneration, so the ventricles in the brain will dilate in Cushing syndrome. All these changes are depending on ACVDBI, and by the way, glucocorticoids have an impact on all what we call the hallmarks of aging, that are the mechanisms, the drivers of the biology of aging. And so the way how it works is that glucocorticoid activation stimulates ACVDBI production by hepatocytes. Circulating ACVDBI then acts on GABA receptors to cause metabolic and immune effects, of course, major immunosuppression, and the extracellular neutralization of ACVDBI and its genetic inactivation can abolish all the facets of glucocorticoid side effects and interrupt a positive and disease favoring feedback loop in the hypothalamo-pituitary adrenal axis. I move to other diseases very shortly. This is one of the most horrible diseases that exists. Unfortunately, it only affects a few dozens of individuals in the world. It is the so-called Hutchinson-Gilford-Progeria syndrome. It's a mutation in lamin A, which is homozygous. The children are born normally, and they develop this premature aging phenotype to die between 12 and 14 years, usually from cardiovascular disease, arteriosclerosis. And so we have a mouse model for this disease, which is a mutation in the protease that matures lamin A, ZMPSTA4. If you just look at the controls versus the mutated mice, you see in the upper row, these are mice with this mutation. You see these kyphosis, and skin defects just by eye. You see this is not a normal mouse, it's a prematurely aging mouse. And if you give the antibody against ACVDBI, we erase this phenotype. And you see, for instance, the signs of kyphosis, the misplacement of the spine are normalized by ACVDBI neutralization. Another disease, which is much more frequent, which is premature aging of the kidney in response to chemotherapy with cisplatin. So you see that in the middle upper panel in red, the cisplatin treatment has led to a reduction of the glomerular filtration rate, the function of the kidney. That is normalized if we neutralize ACVDBI. And you also see that we can quantify by immunohistochemistry senescent cells in the kidney. So in response to cisplatin, you have these tubular epithelial cells that accumulate in the senescent marker that's called P21, or cyclin-dependent kinase inhibitor 1A, that gives these black spots that disappear with neutralization of ACVDBI. And we can move to liver damage that is caused by a high-fat diet combined with the hepatotoxic agent CCL4, which gives you a phenotype of steatosis and fibrosis. You see that the senescence of hepatocytes stimulated by this treatment is abolished by ACVDBI neutralization, as is also reduced fibrosis and the accumulation of liver enzymes such as the transaminases in the blood. And you can move to the premature aging of the heart, stimulated by an anthracycline, doxorubicin, which is given several times weekly for cardiac damage. So this causes cardiac failure that is abolished by ACVDBI neutralization. And again, if you look at the histology of the heart, you see that cardiomyocytes become senescent in response to doxorubicin, but not anymore upon ACVDBI neutralization. And we actually did some single nucleus sequencing of these hearts to identify that actually cardiomyocytes are those cells that are mostly affected by, in the transcriptome by doxorubicin, and that the shifts in the transcriptome are strongly normalized by ACP-dBi neutralization. So just to give you one example, which is BCL-XL that is downregulated by doxorubicin, but upregulated in the presence of ACP-dBi neutralizing antibodies. And just another disease that is problematic, which is osteoarthritis. So here we are dealing with a mouse model in which osteoarthritis is induced by destabilization of the knee articulation. So we cut the median meniscus and the collateral ligament. This mechanical destabilization then causes a local inflammation just in the affected articulation. But you see here a systemic elevation of ACP-dBi plasma levels. So the local disease has a systemic effect. And we can then try to intervene with the disease phenotype by giving a monoclonal antibody against ACP-dBi after the surgical intervention, and see that the signs of osteoarthritis, which is the loss of cartilage, the inflammation, and the destruction of the bone adjacent to the articulation, which can be converted into an index of severity, this osteoarthritis is attenuated by ACP-dBi neutralization. And what we also can do is we can monitor osteoarthritis over time. So these are mice that are monitored at 3, 6, 9, and 12 weeks after surgical intervention. In the sham-operated knee or in the destabilized knee where they develop osteoarthritis. And we can actually then measure the severity of the disease by looking at the edema and hypodense echographic zone that we can quantify. And you see actually that 3 weeks after the treatment, the control and the ACP-dBi treated group are still similar, but as time goes by, the treatment group shows an attenuated osteoarthritis phenotype, which means actually that we can reverse an established pathology. And this is importantly accompanied by a reduction of systemically measured cytokines that are pro-inflammatory in the blood, including interleukin-1 alpha, interleukin-33, and tumor necrosis factor alpha, meaning that the local treatment against a local disease with the antibody against ACP-dBi is able to prevent the systemic manifestations of inflammation. And we move to another disease, an entire group of diseases which is called cancer, although these are molecularly different etiologies. So here we are looking at a model of memory carcinogenesis in which we give progesterone together with a DNA-damaging agent to mice that will, over time, the experiments last a year, develop breast cancer. And you see that if we knock out ACP-dBi in those mice, the progression of the tumors is slowed down. And we can see this also in easier experiments, and we just inject breast cancer cell lines into the memory footpad orthotopically to see that there is tumor growth, the black line, that can be reduced either by the neutralization of ACP-dBi or the neutralization of PD-1, which is a famous target in cancer immunotherapy. And the combination of both actually is synergistic in this effect. And here's a model of lung cancer that is induced by a tobacco antigen, tobacco carcinogen, which is urethane. Urethane, after a long latency of 9 months, induces these black lesions, which are cancers in the lung, that are reduced in the frequency after the stimulation of autoantibodies neutralizing ACVDBI, the blue group compared to the black group. And if we just inject autotopically a lung cancer cell line into the thorax and see these tumors form, then we see that the neutralization of ACVDBI slows down tumor progression over time. And actually, if we do exactly the same experiment in immunodeficient mice that lack T cells, there's no effect of the ACVDBI neutralizing antibody, meaning that this effect is depending on the immune system. And so we can go on and do the experiment slightly differently. So here we wait the cancers to manifest. They are present in all the mice at the same size. And then we start the treatment with ACVDBI neutralization alone, with PD-1 blockade alone, or the combination. So you see here nicely this additive, perhaps synergistic interaction between ACVDBI neutralization and cancer immunotherapy that allows for optimal survival of the mice and optimal control of the tumors. And yet another model of cancer, hepatocellular carcinoma, induced by diet. So it's a high McDonald's-style, high-fat diet that is combined with CCL4. And you see that the largest tumor size is reduced, as well as the average tumor size, by ACVDBI neutralization in this model. And we can now look for the mechanism of this effect and do some spatially resolved transcriptomics to distinguish in these models the area of the liver that is apparently normal and the area that is tumorous. So we distinguish non-tumor from tumor. And we see even that in the apparently normal liver already, the neutralization of ACVDBI reduces the expression of genes that we qualify as immunosuppressive and enhances the expression of genes that we qualify as immunostimulatory. So there's an effect on the local immune system that, again, we can exploit by combining ACVDBI neutralization in this model with PD-1 blockade to show that this is optimally efficient to reduce the weight, the number of tumors, and to guarantee the survival of the mice. And when we then look at the composition of the immune infiltrate by high-dimensional cytometry, you see that the inhibition of ACVDBI alone is already sufficient to enhance the presence of T cells in the liver and to reduce that of regulatory T cells so that as a proxy of local immunosurveillance, the ratio of CD8 positive T cells over regulatory T cells is improved in response to ACVDBI neutralization. And we can do similarly more complicated experiments on the tumor immune infiltrate. This is in the lung, where you see that by T lymphocyte-specific single-cell RNA-seq, we can distinguish 9 different T cell subtypes. And if we zoom on them, we see that the frequency of regulatory T cells is reduced, while that of non-exhausted, activated cytotoxic T lymphocytes is enhanced by ACVDBI neutralization. It's always the same immunostimulatory effect that has this anti-cancer activity. And now if we zoom on all the genes in the 9 subpopulations of T cells, there are very few that are common to all the subpopulations. One among 4 genes that is upregulated is lamin A. Remember, lamin A, the gene that is mutated in Hutchinson-Gilford-Progeroid syndrome. And so it took us just a year and a half to do the experiment, so to knock out lamin A in T cells, and to show that if we consider the controls which have lamin A in T cells in green that respond nicely to cancer immunotherapy combined with ACB-DBI neutralization, this therapeutic effect is largely lost after removal of lamin A from the system, meaning that this is actually the mode of action. And so, to summarize, it appears that ACBP-DBI can be classified as a gerogene, so it's a gene that favors aging. And this is based on the fact that the ACB-DBI contributes to all sorts of age-associated diseases, including cancer, cardiovascular disease, renal disease, metabolic syndrome. I told about Cushing's syndrome, but it's also true for other instances of metabolic syndrome induced by high-fat diet. Sarcopenia, we have the example of osteoarticular diseases, osteoarthritis, osteoporosis, respiratory diseases, and local systemic acute and chronic inflammation. And the mode of action of ACB-DBI seems to be intertwined with other gerogenes, including lamin A. So there's a final twist to my story. So I told you that one of the names of ACB-DBI is endozepine, the endogenous equivalent of benzodiazepines. And that's because benzodiazepines, exemplified here by diazepam, and ACB-DBI act exactly on the same binding site of the benzodiazepine receptor or GABA-A receptor with the same effects. So both of them are positive allosteric modulators that change the threshold for the tool agonist of this receptor, which is GABA, gamma-aminobutyric acid, an inhibitory neurotransmitter that also exists outside of the brain and has multiple signaling functions. So this is just an example to show that ACB-DBI and diazepam have the same effects in mouse models. So here we're dealing again with a model of tumor growth that is treated with chemoimmunotherapy. It improves its response when we inhibit ACB-DBI, but this advantage is lost when we inject diazepam, even at low doses into mice. So the mice don't sleep. They behave apparently normal after intraperitoneal injection of diazepam, and there's immunosuppression. So we recapitulated this in patients. This is an example of non-small cell lung cancers that are under PD-1 blockade in our hospital in Villejuif, in the outskirts of Paris. And you see that those patients who are taking benzodiazepines during immunotherapy, the orange line compared to the blue controls that don't take benzodiazepines, have a shorter overall survival. So they don't respond to immunotherapy. We have recapitulated this result in 3 different cohorts, up to 32,000 patients in the French social security system. There's also a Japanese group that has now shown the same thing. Immunotherapy doesn't work due to immunosuppression by benzodiazepines. And if you look at the literature, which is involving now cohorts of millions of people, it turns actually out that ACB-DBI, that benzodiazepine use or abuse is coupled to the increase in the incidence of cancers, including lung cancer. And there's also a very nice Swedish cohort in which they found, in a country of 8 million adults, 700,000 people using or abusing benzodiazepines on a regular basis. It's very broadly used in the Western countries, and showing that those people who take benzodiazepines are systematically higher risk of developing serious infections that require medical treatment. And we can go further and look at other cohorts to discover that benzodiazepines are coupled to an increased incidence of all sorts of cardiovascular diseases and events, and even the acceleration of dementia and neurodegeneration, suggesting that we actually can say that benzodiazepines induce iatrogenic aging. And so the mechanism of this is that benzodiazepines and ACB-DBI acting on the same receptor, the GABA receptor, inhibit autophagy, induce metabolic syndrome, stimulate senescence and inflammation, and cause immunosuppression, which explains then this vast array of age-related diseases that are the consequence of this pathway. And what we have to do in the healthcare system is, of course, to prescribe benzodiazepines. We have to prescribe glucocorticoids whenever possible, because they are the strongest inducer for ACB-DBI that we can imagine. And we may also try to inhibit ACB-DBI itself with antibodies, or we can try to get rid of the natural receptor agonist, which is GABA, which is produced, for instance, by certain types of malignant tumor cells, but also by the gut microbiota. And we are working on this very actively. And so you have remember this scheme of the different strata and the health characteristics, the hallmarks of health that communicate in their perturbations. We think that maybe ACB-DBI is one of these major communication signals that links local perturbations to systemic disease. Now, I get this question all the time: what can I do to reduce my ACB-DBI levels? And so I cannot give the answer, but there are statistical correlations in an Icelandic cohort of 26,000 patients or individuals, and the UK Biobank with 55,000 individuals, that show that the better your education is, if you don't smoke, if you have a regular physical activity and muscle but little fat, eat a balanced diet, and have high vitamin D levels, you have low ACB-DBI levels, suggesting that a healthy lifestyle is linked to a reduced concentration of this age-accelerating hormone. And with this, I thank you for your attention. I have to mention that this work has been done by over 10 years by an average of 12 persons dedicated 100% to this study. So it's 120 life years of work that has been accomplished by my team in collaboration with many scientists all over the world, because science is always a collective adventure. Thank you so much for your attention.
Thank you, Professor Kromer, for a truly illuminating talk and giving the entire context of ACB. Looks like we are very close to...
The microphone is off.
Thank you for a very illuminating talk and giving, I think, as you said, this is on? I think on the dot ACBP could be a game changer, from an early prognosis to also looking at what would be the severity of the disease that might happen. So I'm sure we can have one or two questions or comments. Professor.
Professor Guido, that was a brilliant talk, absolutely brilliant. So 2 questions I have in mind. Number 1 is, if you discover the pill, whenever that happens, so that should be the panacea for every human disease, because it's all rooted in what you have described, the ACPP-DBI. My last question is, I'm sure you're exploring any connection with dysbiosis and the level of this hormone, because we are so much focusing on human disease and dysbiosis.
Okay, so I will start with the second question, which was easier. So we have looked at the capacity of eubacteria and yeast species in the gut to produce ACP-DBI equivalents. And we have not found strong evidence that the microbiota would produce an equivalent of ACP-DBI that would have a capacity to affect the physiology of mice or humans. So there's probably not a direct link between the microbiota and ACP-DBI. There's an indirect link because inflammation of the gastrointestinal tract, including Crohn's disease and colitis ulcerosa, or peritoneal infection after perforation of the gut leads to an increase in ACP-DBI plasma levels. And we can inject E. coli, that is a gut-derived bacterium, intravenously to mimic septic shock in mice and show that this also causes an ACP-DBI increase and an ACP-DBI-dependent pathology. Now, the first question was a little bit more complicated because you put in the word panacea. So we have a lot of mouse models in which ACP-DBI inhibition has positive effects. Of course, we have not studied all diseases, so we cannot say that it's a panacea. We know that the ACP-DBI inhibition is safe for mice kept in a conventional animal facility. So they live longer, which is a very good proof that it is safe. And they show no histological or macroscopic signs of pathology in any organ. Now, the only side effects that we have seen is that as they age, black mice become beige slightly, which is presumably due to the induction of autophagy in keratinocytes that reduce the melanosomes in the cytoplasm. And bleaching creams used by the cosmetic industry are usually autophagy inducers. So it is safe. It is safe in laboratory conditions. We have some. This is a shock. It's a stress hormone, so it has a physiological function in appetite control, and so we suspect that in the wildlife of mice, when food supply is inconstant, and there is cold and heat stress, predators, and so on, the absence of ACP-DBI would be a disadvantage. And that's actually the reason why it has been phylogenetically conserved through all the speciations of animals and mammals. Well, fortunately, humans are living like mice in luxury conditions in the Western world, and now in India, for the middle class at least as well. And so, presumably, ACP-DBI inhibition would be safe in humans in standard conditions as well, but maybe not in conditions that would correspond to wildlife.
2 questions.
Yes.
It's fascinating that you proposed ACPP as a magic bullet solution for most diseases, but ACPP is a natural homeostatic regulator of aging in biological systems. So 2 questions. ACPP intracellular levels are fairly high, micromolar, but extracellular levels are low, nanomolar. So when we deplete ACPP circulating system, it's possible that the intracellular levels get pumped up. So then would this ACPP attenuation therapy becomes transient therapy? That's question 1. Question 2, are there any large-scale clinical trials going on ACPP attenuation? These are the 2 questions. Thank you.
Okay, so I start with the complicated first question, which was using the word miraculous or magic. So I'm not claiming that it would be a magic pill. It's my disclaimer. Okay. We don't know. So there are specific diseases, and we will have to find carefully which is the best indication to do a first clinical trial that we haven't done yet because we have not yet manufactured a GMP-compatible lot of the neutralizing antibody that we have produced against human and mouse ACBDBI. So we have a cross-reactive antibody that we can test in different animal species, including in cynomolgus monkeys, that we are preparing for future clinical trials, but this is not done yet. So you asked also a question on concentrations. Actually, you are absolutely right. So the concentrations of ACBDBI in the circulation are in the range of 10 to 15 nanogram per ml plasma. And the concentration of ACBDBI within cells is much higher, which, by the way, is led us to the calculation that in a mouse, destruction of as little as 3 to 5 milligrams of liver tissue, which even for a mouse is nothing, is sufficient to double the concentration of ACBDBI plasma levels. So small disease processes locally can enhance ACBDBI plasma levels systemically because of that discrepancy between intracellular and extracellular plasma levels. And so if you want to neutralize ACBDBI in the extracellular space, we certainly will have to titrate the amount of antagonist or antibody against the ACBDBI as a function of this extracellular concentration.
Thank you.
So it was a fascinating presentation. The question I have is, in all the disease contests, you elegantly showed that inhibition of extracellular, sort of extension of the previous question. Have you thought about degrading or inhibiting intracellular? Is it supported by conditional knockout studies? That's question number one. Second, you mostly looked at combination with anti-PD-1. Have you also looked at, say, other T-cell targeted therapy, whether it's CAR-T or whether it's a T-cell engager? What are the data that you would anticipate?
Okay, so the first question was about intracellular versus extracellular ACBDBI. So the phenotype in the disease models that we studied of the neutralizing antibodies or the total knockout of ACBDBI by inducible by tamoxifen is the same. So we don't think that the intracellular pool of ACBDBI has a major function in these diseases unless it is some specific situation. So there's a literature showing that intracellular ACBDBI in glioblastoma favors fatty acid oxidation and fitness of the tumor cells. But in the liver model, in the lung model, in the breast cancer model, and the fibrosarcoma models that we studied, only 4 so far, the role of the intracellular ACBDBI is probably this negligible, which doesn't say still that the ACBDBI that comes from a tumor can be from intracellular origin, of course, but the intracellular function is not important, as far as we can say. And the other question was about synergism with other immunotherapies and PD-1 blockade. So I have to say that we only tested immunogenic chemotherapy, which is our specialty, and immunotherapy with PD-1 blockade or PD-L1 blockade, or the combination of chemotherapy and immunotherapy, so chemoimmunotherapy, but we have not looked at CAR T cells or T-cell engagers or other modalities of immunotherapy so far. What we can say is that natural immunosurveillance, which is not depending on immunotherapy, is also controlled by the levels of ACBDBI. And actually, we just published last week or so a paper in PNAS showing that glucocorticoid-induced immunosuppression that, of course, kills immunosurveillance and causes a flare-up of tumors. That immunosuppression is depending on ACV-DPI as well, even in the absence of immunotherapy. So presumably it is a very vast effect that transcends the PD-1, PD-L1 pathway.
Thank you, Professor Kromer. Let us give a big round of applause. And
I request Mrs. Deepti to kindly present the memento to Professor Kromer. May I request you to come back on stage?
Thank you for the surprise.
Thank you very much, Professor, and thank you, Mrs. Deepti. Thank you. We'll now move on to our next distinguished speaker. And it is indeed an honor to introduce Dr. Reddy, founder and chairman of Continental Hospitals. A distinguished physician specializing in gastroenterology, liver disease, and transplantation, Dr. Reddy received his training at the Baylor College of Medicine in Houston, Texas. He is the founder of Digestive and Liver Disease Consultants and opened one of the largest gastroenterology and liver clinics in Houston. Dr. Reddy has held prestigious positions, including president of the Houston Academy of Medicine and chair of Hospital Council Chiefs of Staff, overseeing over 80 hospitals. In 2013, he established Continental Hospitals with a vision to deliver world-class healthcare rooted in compassion, innovation, and excellence. He is recognized as a champion of empathetic patient care and is the recipient of several awards and accolades. Dr. Reddy has been recognized as 2006 America's Top Physicians by Consumers Research Council of America. He has also been in the top physicians in the United States by the Newsweek magazine for 2 consecutive years. May I now request Dr. Reddy to reflect on his treasured memories of Dr. Anji Reddy.
Good morning. What a wonderful Sunday morning sermon. You know, we all go for Sunday morning often for religious events in the West, going to a church and listening to, a fantastic preacher to really regenerate ourselves and get ready for the following week. But this was something incredible. Coming on a Sunday morning, listening to a great researcher, a renowned scientist, who has introduced to us optimistically there is something more coming in management of human disease, and simplified so well at the end of the day. And truly, it befits for us to celebrate the man in whose honor that we all assembled today, Dr. Anji Reddy Garu, and the memorial lecture that befits Dr. Reddy's ethos, his dreams, his persuasion of science, what he lived for, and how he drove his life, establishing one of the largest companies, but nevertheless, not just to manufacture medicines or drugs. As it clearly illustrates at the bottom, a man who wanted to foray into research and for India, design, invent, and deliver absolutely, a drug from bottoms up. His Life Sciences Institute, his foundation, the Origin and Origin Oncology, all of them remain a testament, a tribute to his persuasive and indulgence in the world of science and something to happen in terms of helping human disease. It's my deep honor today and absolutely a privilege to reflect some of my encounters or associations with Dr. Reddy over a period of 2 decades plus. Dr. Anji Reddy's family was known to my parents, particularly my mother, Swarajamanti, who was very nicely used to be talked about by mother. And of course, Prasad's parents were also equally close. So this relationship goes back to several decades. And when Dr. Reddy's was opened in 1984, I was already in the United States of America the previous year, in 1983. There are 2 historical milestones. I'll come to the latter, which I can never forget, his anniversary, and I will outline what is that. So thank you, family. Thank you, Satish. Thank you, Deepthi. Thank you, Prasad. Thank you, Anu, and thank you, Shravya, and all the rest of the grandchildren for giving me this opportunity to participate and speak about what I know of Dr. Reddy. I met Dr. Reddy, as I mentioned, at least a dozen times, both in Hyderabad, on my visits to Hyderabad, as well as in the United States of America. So one of my first meetings was at my home. We are hosting a Medicity CME event that was happening in Hyderabad with almost 90% of the faculty which was visiting with the US. During that time, Hyderabad was declared, or state of Andhra Pradesh was a dry spell, means nobody can drink alcohol. And all the speakers who visited the faculty were truly agitated. But I nevertheless hosted a dinner. I was here just for a week. And then I live on the same street as a CM of Andhra Pradesh who declared, no alcohol. So we somewhat bootlegged alcohol for all the visiting faculty, and we made sure that everybody had a nice time, including Dr. Reddy that evening. And because so many people were there who wanted to speak to Dr. Reddy, I did not have really much time. Nevertheless, I told him, sometimes I addressed him as uncle, sometimes I addressed him as sir. I said I was finishing up my gastroenterology fellowship at the Baylor College of Medicine, and this is my dream, this is what I want to do. And he was extraordinarily encouraging, at that point in time, although it was a very brief meeting. My next meeting was when I visited somewhere in the early 1990s. It was in Taj Krishna, and I remember in the corner, and nobody was sitting around him. And I did ask somebody, where could I meet Dr. Reddy? So I said, this will be the time if you visit. So I really went down, sat with him, almost spent about 45 minutes. And during that time, I explained to him, what I was doing and my interest in clinical research, which I was associated with during that time, even including my days of residency, where they gave me a 1 year instead of doing a 3 years residency. My program director was kind enough, because I had so much of interest in research. Hey, Guru, I'll give you a year of reprieve from your clinical medicine. You can go to the bench research. So that was the only year of my career where I spent with test tubes and the animal labs. But then I realized my passion for clinical medicine was extraordinary, and I cannot be a bench researcher. And let me pursue clinical research. So during that visitation with Dr. Reddy, who was so extraordinarily inspirational in terms of what he was doing, and his interest was, he did tell me, if India can come out with a medication, and my pursuit for research, which I'm working on, by which time he has not yet established the clinical R&D divisions in the United States of America. Look at his life story. A man who was born so simply in a village, goes on to graduate high school with no support, and pursues a dream to get 2 undergraduate degrees, a Bachelor of Science and a Bachelor of Technology, and subsequently moves to Mumbai, where he finishes his master's and then pursues a PhD in chemical engineering. At that point in time, I bet what went through his mind, and he fully realized perhaps the future is not something we enter. The future is something we create. And he did create the future. He did create the future for the masses to the millions and the billions that we will quickly go through. So for all of us have many opportunities in life, many chances in life. I believe Dr. Reddy has, his options remained, basically the 3 C's at the end of the day. He had the courage, he had the vision to, pursue what he wanted to do at the end of the day with clarity, and he wanted to truly take a chance. So persons who do not take chance in life and do not have courage in life, it is very difficult for them to realize their dreams, and simply dreaming would not help. So he was endowed with those things, without any big display that he was that. He was soft-spoken, keenly watched, and he said to me that you should believe in yourself, work hard, and pursue your dreams. At one of these evenings, it was very clear to me, how could this man become so successful? Was there anybody behind him? But ladies and gentlemen, let me tell you, his success was not the sleight of a magician's hand. His success was not the tricks of Horatio Alter. His success was not any dexterity of any Houdini lurking across the world. His success was basically self-made. He was a man with grit. He was a man with a purpose. And he was a man with a vision who otherwise would not have established, would not have come out of a premier public company where he was working at IDPL and had the dream at a young age of his late 30s to start a company with nobody's support, and successively within 2 companies in 1984 come to establish DRL. Bold in invention, humble in service, Dr. Reddy built a pharma legacy where innovation always met with compassion. India then was importing your APIs or whatever was to deal with pharma chemicals. Dr. Reddy transformed within a very short time as an export industry. Extraordinary, laudable. He's widely credited with mentoring an entire generation of entrepreneurs who went on to build their own pharma companies in this city and state, turning Hyderabad into the bulk drug capital of India. Now, you will find it very interesting on this particular journey of Dr. Reddy. How could somebody working for him, how could an employee of him live the company, exactly embrace and emulate what he was doing, and this man never prevented, never exercised a no competition clause, never went and knocked on the courts, never sued anybody. But interestingly, he helped these very people where the help was needed, whatever they was needed, in terms of mentorship, in terms of buying the products, in terms of giving them what they needed. I do not think so, or any one of you will find such an example, because most of them went on to become the billionaires of this city and state. That is extraordinary fact of his journey. And his role in creating pharma billionaires was simply rooted in an entrepreneurial culture that he believed in. And he stuck to that for the rest of his life. His generosity and guidance, truly, they all became something because of his extraordinary generosity and guidance and the legacy of his laboratories. No day he compromised. And they all succeeded in emulating that. Dr. Reddy, all of us know, is often referred to as the Bhishma Pitama of the Indian pharma industry. His legacy is deemed responsible for creating substantial wealth and employment in this sector, in this country, and subsequently abroad. He didn't just run a pharmaceutical company. In fact, if you talk about how these employees and rest of them became such billionaires established in the pharma industry, to me it looks like he ran a biological experiment. He took a handful of employees, injected them with a growth serum of ambition, and then watched in horror and pride as they underwent mitosis. Now, every time he looks at Hyderabad's skyline, he's not looking at a city. He's looking at his own extended family tree, where every branch is trying to sell him the same aspirin for half the price. That was an incredible story. Now, Dr. Reddy led by lifting others. He never stood tall for himself. It was throughout his journey. His greatest product was not a drug, but the leaders that he trained, the generation of scientists that he trained, the managers that he trained, the entrepreneurs that he trained. His vision remained never blurred at any point in time. It was always razor sharp. The successes of his bulk drugs, we talked about this in San Diego at one of the RP conventions. The successes of his bulk drugs or the successes of his APIs or generics or formulations where he had the challenge to create the new formulations or new drugs or drugs that he was making by reverse engineering, did not just satisfy him. Yes, he did this for the masses. He was very proud about this. A product that could be sold just for pennies in the Western world when they did not have access to medicine. This pioneer from India, made this happen for humanity. But he was passionate to invent a drug. He did tell that. He did tell to me when we were conversing, India should be one of the countries that should innovate and have its own drug. And he didn't want to be just merely be labeled as a generic manufacturer. This burning desire certainly went on to start the R&D facilities in the early 1990s, probably the only Indian pharmaceutical company, and was the first to its credit to do that in the US. During a meeting in American Association of Physicians of India in San Diego, Dr. Reddy used to attend that very regularly. We were both having lunch or a sandwich, and he was invited to give a scientific lecture. So I asked him, sir, how do the American pharma giants view you as Dr. Reddy's labs? Do they find a threat? Through my recollection, what I penned here, he mentioned what I summarized here, the rising power in the Indian pharmaceutical industry for low-cost drugs was indeed a concern for the pharma giants. But nevertheless, in the 2000s, the pharma giants viewed Dr. Reddy and DRL as a disruptive and innovative face rather than just a generic copycat. When we talk about today, India's progress in sciences, technology, manufacturing via disruption, via innovation, Dr. Reddy was already there. He was there a long time back. So that was disruption, what he did. Reverse engineering, making drugs for humanity and the masses, was truly an innovative, brilliant idea. And this was recognized by the giant companies, shifting from viewing Dr. Reddy as a threat to a formidable partner, truly a formidable partner. And we are not saying this. He has proved it, what has happened subsequently. Some of the key milestones, those of you remember what has happened. The Pfizer, multi-billion-dollar drug company, sued DRL against a patent infringement lawsuit. and Dr. Reddy has successfully won that. And that was an eye-opener. With the winning of that lawsuit, later on, other pharma companies like GSK and others wanted to partner with Dr. Reddy, which happened in the later part of 2000s. And then starting of his R&D facilities also showed to these pharma giants that he was not there just to do low-cost drug manufacturing. He was a serious player in the industry. And these were the pearls, these were the gems of this man. How could one human being, taking 1 billion people on his shoulder, prove to a well-established pharma industry abroad and make them, give them, have him be given the same stand on the same platform? Hence, the first decade of the 21st century for Dr. Reddy was earning the respect on the global platform as a partner of choice. Subsequently, with his buying of the beta pharma industry, he also forayed into Europe very aggressively. That was the chapter of opening into the European world. Now, this is a real interesting segment that I went through. When he listed DRL in the year 2001, first Indian company which was listed for $132 million. And the first drug was fluoxetine. Prozac everybody was using like candy in America in those days. It was not the PPI, but Prozac, the antidepressant drug, which was so widely used. And with Dr. Reddy's introduction, certainly the price came down. And me as a gastroenterologist, we were interested in when we introduced the Prilosec version, the generic version, the omeprazole, the 10 and 20 milligrams, the long-acting release, which was then being made by AstraZeneca. AstraZeneca again sued him, basically that this cannot be done. But nevertheless, Dr. Reddy proved just in a span of 6 to 9 months, this is a different molecule, this is long-acting, and the judge clearly pronounced there was no issue, released in the market. So 2005, when the first PPI was introduced. So my patients, as a gastroenterologist seeing them, they used to ask me, they went on almost every month 3 or 4 questions for the next years or 2 years. Are you kin to Dr. Reddy was one of the questions. The other question was, do you own Dr. Reddy's stock? The third question was, would you recommend to invest in Dr. Reddy's stock? Because when omeprazole was introduced, it was a blockbuster. The stock price went up by 25 to 30%. So the Americans who were invested there, they were really enjoying it. Those who were retirees, semi-retirees, I didn't know that, that Dr. Reddy's was even listed then. But I did take that advice and sneakily had my guy invest and buy Dr. Reddy's at that point in time. So depending on the tone and the mood of the patient, I tailored my answers. And those of them who asked me, am I kin to Dr. Reddy? I would have been very proud to say, yes, Reddy, whatever it is, somewhere you're connected. But then I was worried, just in case if the stock price was down recently, this guy may not be really happy. So I better refrain from answering. Number 2 question, would you recommend investment? If I said I own the stock, then they would have surmised when I'm prescribing omeprazole that I am really interested in pushing the drug. So I was very careful in answering that question. Lastly, would you recommend investing? I stayed away even from that advice because I didn't want them to surmise that in case if something happens to the stock or the share, that they would not come around. Because as such, US is somewhat of a litigious society. And they would say, I went to Dr. Reddy for my GERD, and this man recommended me to buy shares of the company. And lo and behold, those things can happen there. So Dr. Reddy's social conscience, on another aspect, if you reflect of his character, he felt business was service, truly. And it was reflected throughout his journey. He said corporate success should never translate into pure profits, but it should be there for social benefit. And he has done that and proven repeatedly. hence his foundation's work. In conclusion, Dr. Reddy was a great leader, but we also celebrate a life that continues to shape how we work and why we work. Let us live here this afternoon recommitted to the values he taught us, to put patients first, to pursue innovation with integrity, and to mentor the next generation who will carry forward his important work. As a physician, as a researcher, as an FDA researcher, continue to associate with research in America and also in Hyderabad. You know, I feel what Hippocrates said once, if you love medicine, you love humanity. And those are the ideals of Dr. Anji Reddy. I'm so proud to be here this afternoon to talk about him and few of my encounters with him. Thank you all. Appreciate it.
Thank you very much. Yeah, bless you to stay on stage. Thank you very much, Dr. Guru Reddy. I think you have really captured the heart and the pulse of Dr. Anji Reddy, talking about how he encouraged you to realize your potential, to dream big, and always have the courage, clarity, take a chance, and most importantly, how innovation is always met with compassion. And I think that also resonates back to the quote that Professor Cromer referred to, that he had on his, was on his whiteboard you referred on whiteboard for a long time, about how this is really for the benefit of humanity. And thank you so much. And this has all been, in fact, embedded in the DNA of our organization, and we, live it every single day that, we work at Dr. Reddy's. And I would now request Mrs. Anuradha Prasad to kindly present the memento to Dr. Guru Reddy. Thank you very much. Thank you once again. Yeah. May I now request Mr. Satish Reddy to deliver the concluding remarks.
A very good afternoon, everybody. It's past 12 o'clock, so good afternoon to all of you. Thank you for being here today on a Sunday once again on this very, very special occasion of the fourth Dr. Anji Reddy Memorial Lecture. Every year we try our best to improve from the previous year. I hope there's been no shortcomings for this year. And we are especially fortunate that Professor Guido Kramer has come in all the way. I know he must have been very tired. He came in early yesterday, and then we had a round table at the Institute, and he was very, very enthusiastically participating in it, along with several members who were part of that round table, some of whom are also here today. I just wanted to acknowledge about the round table, because again, just to give a background of the round table, just a day before the memorial lecture, we conduct a round table on issues which are very important for the country. When we first started this 3 years ago, it was on emerging frameworks for innovation in this country. The second one last year was about public health. Again, we invite eminent people from across the country, from various fields of expertise, to also share their insights, their thoughts, and then we publish a white paper out of it. And the moderation is done by the very well-known publication Sightline, which comes out with publications such as Script, Pink Sheet. Some of you will be very familiar with those publications, which is distributed for life sciences companies across the world. We make them aware of the current situation in the country on topics of interest. So this year it was about translational research. And we had the good fortune of several people who have participated in it. Dr. B.J. Rao is here today. He was also part of that roundtable, Vice Chancellor of University of Hyderabad. He just retired, if I'm not mistaken, today or yesterday? Yesterday. So he made the time to be with us for that. We also had Shiv Kumar, who's the CEO of ANRF, Anusandhan Research, National Research Fund, which is in the limelight of late because not only because of the size of the fund, but it is a fund that's been put together by the government of India to encourage innovation in this country. So he was also part of that. Dr. Satya Dash from IKP, he's here somewhere here. Oh, there he is. So he's also here with his wife and family, his daughter also here. So he was also part of the roundtable yesterday, also sharing his insights from the Knowledge Park and what he represents from his past experience. Also, Dr. Sarkar from Continental Hospitals, he's also here. I think he's here with his wife. Thank you for being there yesterday. Dr. Soumya Swaminathan and Dr. Ram Gopal Rao from BITS Pilani, they were also joined us online. They couldn't be here in person, but they joined online. Murli from Origin was also part of the discussion. Also Professor Guido, he was also part of the roundtable. So we had a great discussion yesterday on translational research. A white paper also will come out of that the proceedings, and that's something we'll distribute to all of you. So that was one of the things. I was also reflecting that, again, looking at the audience, the person who would have been most happiest in this room was Dr. Anji Reddy himself, if he was in this room. He was always, like Prasad mentioned earlier, very comfortable, very, very comfortable in the presence of scientists, several of whom are present here today. He's also very excited to be in the company of friends, some of whom are also here today. And he would have just enjoyed the whole interaction, listening to Professor Guido give his lecture, because he was a man of intellectual curiosity. He always wanted to keep updated about scientific developments, interact with scientists, and that's what gave him all the excitement. And that's something we dearly miss him for that. I wanted to specifically thank the people who organized this event, starting with Srinivas. Every year he takes great pains putting it together, inviting the speakers, looking after all the arrangements, also making sure that the roundtable is organized at the Institute. He spends extra effort. Again, once again, thank you, Srinivas, for this. Also, people from Dr. Reddy's, Dinesh, people from Studio 5B, Boris and his team, who also have taken great pains to put all the arrangements together. Raju, Madhumita from our offices, and Priya also from corporate communications. Srividya, who's always ably guiding us through this event by conducting it. Several others who have participated to make sure that the event goes around successfully. So before I conclude, I just wanted to also remember Dr. Reddy, on this occasion, more than anything else, for the institutions that he built, especially Dr. Reddy, the company. Dr. Reddy is the company. That's something which was very close to his heart, and the very fond recollections Dr. Guru Reddy talked about, not only about his personal interactions, but also about the company, the way it was built, not only the company, but also people who have gone out of the company to also build big companies, and also very entrepreneurial what they have done. This is all a testament to the success. But more to me especially, which is always heartening, is the institutions, besides Dr. Reddy's that he has built, are also what he did on the philanthropic front. Dr. Reddy's Foundation, there's also Nandi Foundation, Manoj is here. Nice Foundation, Dr. Padmanabh Reddy is here. Especially if you were to recollect at what stage all these institutions are at this today, I think it would have swelled his heart with pride, because from what was seeded very early by him to the kind of institutions they have become. Dr. Reddy's you're familiar with, so I don't have to talk about, it's like to the heights that it has reached. But if you see Nandi Foundation, for example, especially Araku coffee, I always keep remembering it because the farmers of Araku Valley, the kind of income that has risen for them, because it was one of those Naxalite-infested areas when the whole work started by Manoj and his team at that time. But what always, if you were to reflect today, if you hear about Araku, you also would know about Araku, the coffee, the brand. This is something, starting in 2016 when the first cafe was opened in Paris. Now we have 3 cafes there in Paris, the heart of the coffee capital in Europe. And then we have 2 in Bangalore, 1 in Mumbai. And interestingly, we'll also be starting one, hopefully sometime this year, in New York. Right, so that's where it has come, because it's not about just the cafes and the enterprise that have been started by the trustees of Nandi Foundation, but it's also about what it does to build that brand and also ultimately benefit the farmers of Araku. I think that's something which is of tremendous importance. Also, the nice foundation. Since the time it was started, the kind of work that it does, also through the foundation all across the country, in terms of what it does for training, skilling, and also benefiting the people, the really poor people of various regions across Andhra Pradesh, Meghalaya, various parts where the foundation has done the work. It has left an imprint, which I think is something all of us are always proud of. Dr. Reddy's foundation itself has also done great work, and it continues to keep building on the legacy that he has left behind to make sure that we also reach greater heights, not only just in the field of science that he has made a big name through the innovation that happens through Origin, but also through the institutions that he has built to also impact lives all across the country. So that's something all of us are proud of. So once again, thank you all for making time on a Sunday evening to be here with us. We're extremely fortunate to have you all with us here today, and we'll continue this legacy. We'll continue the lectures, the memorial lectures that we have done. Professor Guido, thank you once again for being here. Professor Guru Reddy for delivering remembrances of Dr. Reddy, and we hope to continue this good work. Thank you very much. Thank you so much.
Thank you very much, Mr. Satish, and thank you all for being here. And once again, thank you to our distinguished speakers, Professor Giro Kramer and Dr. Guru Reddy, for honoring this occasion with a lot of insightful talks. And just want to invite you all, if you haven't had a chance, please go through the journey wall at the back to see how we have actually grown through the years with Dr. Kallam Anji Reddy. And we invite you all for a lunch, which is being served outside. So please do join us and stay for the lunch. Thank you very much, and thank you, one and all.